In prokaryotes, it is well known that variation in sequence upstream of an open reading frame has a big effect on translation. Modifying this region of a part is usually the best place to alter expression levels in the cell. Modeling the relationship between sequences and expression level can be handled in multiple ways. But in this paper, relative expression levels are predicted using models that include notions of RNA secondary structure. In the Salis approach, the mRNA molecule is modeled according to distinct attributes known to affect gene expression. Just as in transcription, the initiation of translation begins with a simple binding equilibrium between the Shine-Delgarno region of the mRNA and the 30S subunit of the ribosome. Salis uses a biophysical model to correlate this binding equilibrium with structural properties of the RNA sequence. In this model, for any one RNA sequence, the molecule exists in one of two states. Either the Shine-Delgarno region is available for binding, or it is occluded by a hairpin. If we want to account for the fraction of mRNAs that are competent for initiation, we need to model the fraction of these species quantitatively. The stability of any secondary structures in the 5' UTR that involve the Shine-Delgarno sequence result in this occluded state, and the stability of that complex can be calculated pretty accurately using M-fold models. Salis breaks apart four different aspects of the sequence: the stability of the upstream region called the standby site, the affinity of the Shine-Delgarno region for the 30S subunit, how far apart the start codon and Shine-Delgarno are from one another, and whether it has an ATG or GTG start. And they combine these parameters to get a total energy difference between the occluded and open states. This term describing the affinity of the Shine-Delgarno sequence for the rRNA is based on the stability of the Watson-Crick duplex in this region of the molecule. The models for calculating these energies are well established, and this attribute is believed to be the dominant determinant of relative gene expression levels. The delta G start has only two potential values, one if the start codon is GTG and another if it is ATG. This spacing term deals with the distance of the Shine-Delgarno sequence from the start codon. Typically, the Shine-Delgarno is placed 8 base pairs away from the start codon, so the spacing value has a limited number of integer values, and these were empirically determined. The standby term deals with the stability of the region upstream of the RBS, and this is specific to the sequence of that region. Its value can be estimated by M-fold calculations. Finally, there is the delta G mRNA term. This is the term that estimates the fraction of molecules in which the Shine-Delgarno is hairpinned on itself. They calculate this by taking the lowest energy hit from the M-fold calculation of the entire mRNA structure that involves that region of the sequence. I put stars by the components of this model that are also captured in the part model. The mRNA-rRNA stability is defined by that specific sequence of mRNA in the RBS region of the molecule, and thus this value is captured by the part model. Whether the start codon is GTG or ATG and its distance from the RBS are also captured by the part model. To some degree, the standby energy is captured in the part model in the sense that if these interactions are all local to the sequence upstream of the Shine-Delgarno, they will be independent of the sequence downstream of the start codon. So to some degree, this energy is captured in the part concept as well. The remaining term that distinguishes the occluded and non-occluded populations is definitely not captured in the part concept and accounts for much of the improved accuracy of this model relative to the simple part model. Here I've indicated the URL for the RBS calculator, so you can check it out online. You can try copying and pasting the sequence into it or any other ORF. The claims made about these predictions are that the accuracy is within a factor of 2 for relative translation rate. Note that it does not predict the absolute translation rate, and the actual rate would depend on growth conditions in the specific sequence of your gene. The designs generated by RBS Calculator are 5' UTR sequences. It will not, for example, suggest that you change the codon usage in the first few amino acids of your protein, and sometimes such mutations are exactly what is needed to make a protein express well. The RBS Calculator is the current state-of-the-art in predicting translation rates, but several attributes of the model could be expanded upon to increase its accuracy. For example, the calculator assumes that the translation initiation is under thermodynamic control and thus can be described by delta G's. It is unclear whether a pseudo-equilibrium is ever achieved during translation, and thus may actually be under kinetic control, meaning that it would need to be described in terms of rate constants instead of equilibria. Additionally, there could be higher refinement to the RNA secondary structure calculation. Currently, it only takes into account the single best hit from M-fold. In practice, there could be 10 different structures all similarly populated, and an accurate prediction of the non-occluded fraction could require a description of the partition function for all distinct conformers. The model also only deals with issues of secondary structure and interactions with the ribosome. It does not take into account various proteins in the cell that could bind to and alter the conformation of the mRNA. It also neglects the activity of RNases that could cleave the RNA into different molecules. These represent several ideas that have been proposed for how to optimize this algorithm, and ultimately what features are the right features to accurately model expression levels is something that will have to be demonstrated empirically. The RBS calculator is the first attempt at assembling our biochemical knowledge about translation and encapsulating it in a software design tool, and this approach will continue to improve accuracy as the modeling is refined.